Hello, I am Professor Sims, and in this video, I will discuss blood as it relates to the cardiovascular system. This is the second in a series of 10 lessons held as part of my Anatomy and Physiology 2 online laboratory course. If you're a student currently enrolled in this course, please consult the syllabus in the course Moodle site for assignments, quizzes, due dates, and other course information. In this unit, we will learn about the functions of blood, the cellular and extracellular components of blood, including plasma and the formed elements. We'll identify the formed elements of blood in a smear. We'll identify and explain the physiological basis of blood types, and we will perform and interpret results from a basic blood typing test. Blood plays three main functional roles. Transport is perhaps the most familiar. Blood delivers oxygen from the lungs to the body tissues and carries carbon dioxide back to the lungs to be exhaled. It also distributes nutrients absorbed from the digestive tract, hormones from the endocrine glands, heat to help regulate body temperature, and waste products like urea to the kidneys for excretion. Blood also helps with regulation. It maintains pH balance using buffers. It keeps the body temperature stable by redistributing heat and controls the water content of cells through interactions with plasma proteins and electrolytes. Finally, blood plays a major role in protection because it contains clotting factors that prevent blood loss when vessels are damaged, and it carries immune cells and antibodies that defend against infections and disease. Let's shift to talk about the components and physical characteristics of blood. So an average adult has about 4 to 6 liters of blood, which makes up roughly 8% of your total body weight. The temperature of blood is slightly higher than our normal body temperature, about 100.4 degrees Fahrenheit, which helps us with thermoregulation. And blood is slightly alkaline with a normal pH range between 7.35 and 7.45, which is tightly regulated to support enzyme function and cellular processes. Blood is composed of two major parts, plasma and formed elements. Plasma makes up 55% of the total blood volume. It's straw-colored fluid that's mostly water, but it also contains dissolved substances like plasma proteins, electrolytes, nutrients, hormones, and waste products. These proteins include albumin, which helps maintain blood pressure, globulins, which include antibodies, and fibrinogen, which is essential for clotting. The remaining 45% of blood is made up of formed elements, which include red blood cells, or RBCs, which carry oxygen using the protein hemoglobin. White blood cells, or WBCs, are involved in immune defense against pathogens. Platelets are cell fragments that play a role in blood clotting, and so even though it looks like a single red fluid, blood is a complex tissue made up of many components with specific roles. If you have a look at this table here, it gives a little bit of a summary of the different compositions of blood. So over here on the left, you have plasma, 55%. You have water, salts, electrolytes, plasma proteins, and other substances that are transported by blood, like glucose, fatty acids, amino acids, urea, uric acid, oxygen, CO2, hormones. And then over here, it gives a nice breakdown of the other 45% of your blood, which is going to be your red blood cells, erythrocytes, or RBCs. Those are all the same thing. White blood cells are also called leukocytes, or WBCs. And then within the leukocytes, the WBCs, you have basophils, neutrophils, eosinophil, lymphocytes, and monocytes. We'll talk some more about those shortly. In addition to the composition and the different constituents, make sure that you understand the major functions of plasma and formed elements in blood. The formed elements is the name that's given to all of the cellular and cell fragment components of blood. That means red blood cells, white blood cells, platelets, they're all part of formed elements. And then within the formed elements, we identify subsets called blood cells. Their blood cells have to be fully intact, complete cells, right? So like red blood cells and white blood cells, they each have a membrane, a cytoplasm, and at least in the case of white blood cells, a nucleus. Platelets, on the other hand, are not really cells. They're not true cells. They're cell fragments derived from large bone marrow cells called megakaryocytes. Even though they lack nuclei, they're just fragments. Platelets are still considered formed elements because they circulate in the blood and perform essential functions, in this case, blood clotting. So here's the key distinction to keep in mind. All blood cells are formed elements, but not all formed elements are blood cells, because platelets, while important, are not full cells. Now let's break down the individual formed elements a little bit more, especially how they are going to appear under a microscope. So first you have the red blood cells, the RBCs, erythrocytes. Those are all the same thing with different names. And these are the most numerous formed element. They are going to appear as round biconcave discs that have stain that is a pale pink color. And you'll notice that they have no nucleus, which is one of their most distinguishing features. And those, again, are the cells that carry oxygen. Then you have platelets, and those are cell fragments. They're a lot smaller than red blood cells, and you'll see them scattered in between the red blood cells. And then despite their size, again, remember platelets are really important for blood clotting. Now, the white blood cells, there's fewer of these in number, but they do have a higher variability in their appearance. So here's how you tell the different types of white blood cells apart. Eosinophils have a bilobed nucleus. They're filled with large red-orange granules. They're especially active during allergic reactions and parasitic infections. Neutrophils are multi-lobed. They have a multi-lobed nucleus, typically 3 to 5 lobes. They're pale. They have pale granules in the cytoplasm, and these are the most common type of white blood cells because they're the first responders during an infection. Basophils are pretty easy to spot because they have dark purple granules that often obscure the nucleus. It makes it hard to even see the nucleus. And these are the rarest types of leukocytes or white blood cells, and these are the ones that are involved in inflammatory and allergic responses. Lymphocytes are usually small with a large round nucleus that takes up most of the cell, and it leaves just a little thin rim of pale cytoplasm. And these cells are important for adaptive immunity. And then monocytes are the largest of the white blood cells. They have a kidney shape or a horseshoe shaped nucleus, and these are abundant in terms of the amount of cytoplasm. So the lymphocytes have a really big round nucleus, very small cytoplasm. Monocytes have a lot of cytoplasm, much smaller nucleus. And these are the cells that eventually become macrophages once they are transported into tissues. Let's take a closer look at the red blood cells. One of the defining features of erythrocytes is they have no nucleus, no organelles. And this might seem unusual for a cell, but it creates more space for the cell to carry oxygen and CO2, which is their main function. Their biconcave shape it's kind of like a donut that doesn't have a hole. Like it's squished in the middle, but it doesn't have a hole. And this is functioning as an increased surface area relative to volume, which maximizes the efficiency of gas exchange. And it also helps the cells squeeze through little tiny capillaries without rupturing. Erythrocytes have a lifespan of about 120 days. And then after that, they're typically broken down in the spleen, which acts as a kind of blood cell recycling center. And the surface of red blood cells carries specific molecules called blood group antigens. These are what determine your blood type, AB, or O, and they're critically important for safe blood transfusions. And erythrocytes are packed with hemoglobin. Hemoglobin is a large protein that binds and carries oxygen from the lungs to the tissues, and then it also transports carbon dioxide from the tissues back to the lungs for exhalation. Now let's look at the leukocytes, the white blood cells. These are really important for the immune system. These cells help to defend the body against infection, foreign substances, and abnormal cells. Interest Interestingly, the leukocytes develop from the same stem cells in the bone marrow as red blood cells do, but they take a different developmental path. Unlike red blood cells, white blood cells have a nucleus and organelles, which allow them to perform complex immune functions. However, they do not contain hemoglobin, since their job isn't to transport gases. Based on the presence or absence of visible granules in their cytoplasm when stained and viewed under a microscope, leukocytes can be classified into two main groups. You have granulocytes and agranulocytes. Granulocytes include neutrophils, eosinophils, and basophils, and these cells contain granules filled with enzymes and chemicals that they use to fight infection and mediate inflammation. Agranulocytes consist of monocytes and lymphocytes, and these are the cells that don't have granules, and they have really different roles in the immune system, such as engulfing pathogens or coordinating the immune system responses. Platelets are fragments of much larger cells called megakaryocytes, which reside in the bone marrow, and so they're not complete cells themselves, but they're actually tiny pieces of cells that circulate in the blood. The primary role of platelets is in clot formation, as we've said, during hemostasis, which is the process that stops bleeding when a blood vessel is injured. So when a vessel is damaged, platelets will stick together at the injury site to form a temporary plug, and this plug helps to quickly reduce blood loss. While the platelet plug forms, a more stable clot made of protein called fibrin is created to reinforce the plug and seal the wound. So platelets are important first responders in that they prevent the excessive bleeding and help the body to heal after injury. Now let's get into the concept of blood typing and talk about why it is so important. So on the surface of red blood cells, as we've discussed, there are many different membrane surface antigens, and these are molecules that act like identification tags for the immune system. So the antigens are primarily made of glycoproteins and glycolipids, which are combinations of sugars and proteins or fats. The variety of surface antigens allows blood to be classified into about 24 different blood group systems. And then within each blood group system, several specific blood types vary based on which antigens are present. The most common and medically important of these is the ABO blood group system. This you've heard of. This is the basis for blood typing in transfusions and organ transplants. Type A blood has A antigens on the surface of the red blood cells. The plasma contains anti-B antibodies that would attack B antigens if introduced. Type B blood has B antigens on the red blood cell's surface and carries anti-A antibodies in the plasma. Type AB blood has both A and B antigens on the surface of the red blood cells, but importantly, it does not have any anti-A or anti-B antibodies in the plasma. And because of this, people with type AB blood are known as universal recipients, which means they can receive a blood transfusion from any ABO type without any immune reaction. And then type O blood lacks both A and B antigens on the surface, but its plasma contains both anti-A and anti-B antibodies. So this means that people with type O blood are universal donors because their red blood cells won't be attacked by recipients' antibodies. Let's talk about another important blood group marker called the Rh antigen. The Rh antigen is a protein found on the surface of red blood cells, and if someone has this antigen, they're classified as Rh positive. In the United States, about 85% of people are Rh positive, but if the Rh antigen is absent, the person is considered Rh negative, right? And people with Rh negative blood do not naturally have anti-Rh antibodies. However, they can develop them if they're exposed to Rh positive blood. And this exposure can occur during an unmatched blood transfusion, during pregnancy, or if an Rh negative mother carries an Rh positive baby. The Rh factor is important in blood transfusions and pregnancy care, as mismatches can cause serious immune reactions. Here's how a typical blood typing test will work in the lab. The test uses specific antibodies that react with A, B, and Rh antigens that can be detected on the red blood cells. So using a small drop of a person's blood, you're going to mix 3 separate solutions using 3 different serums. You have anti-A, anti-B, and anti-Rh serum. If the blood clots, which is called agglutinate, if it agglutinates in a particular well with a particular serum, then that means the corresponding antigen is present on the red blood cells. For example, agglutination in the anti-A well of a test, that means the blood has A antigens. If there's no clumping, there's no agglutination, then the antigen is absent. By observing which wells agglutinate and which don't, the blood type can be accurately determined, including whether the person is Rh positive or negative. This figure here shows an example of a sample that has agglutination in anti-A and B. So this is an AB blood type. This is showing no agglutination for A and then positive agglutination for B. So this is a B blood type. You have a type A here where we have clumping in the A well, but nothing in the B well. And then type O is showing no clumping for either one. So for this lab session's pre-lab work, first you want to make sure that you read the blood chapter in the textbook. So that's chapter 18 in the OpenStax book. This will give you a good foundation in blood structure and function. Next, you want to review the introduction and background sections in chapter 2 of the lab manual. These will give you more context and help to explain the goals of the lab. Finally, do the pre-lab exercises numbered 2.1 through 2.4 and use the answers for those to complete the pre-lab quiz on Moodle. After that, for lab exercise 2.1, you're going to be looking at different formed elements of blood in models and simulated slides. So begin by looking at chapter 2 of the lab manual and use the images there to guide you. So figure 2.6 that has the erythrocyte and leukocyte models, you will use those to identify and label erythrocytes, neutrophils, lymphocytes, monocytes, eosinophil, and basophil. Then you will move on to the figure 2.7 where you will be identifying those same cells, but it's on a blood slide, and there's a few more components that you'll be able to see, which include the thrombocytes, plasma, leukocytes, erythrocytes. Specifically, as far as leukocytes, you will be able to see the neutrophils, lymphocytes, monocytes, eosinophils, and basophils again. And then this exercise helps you to become more familiar with the appearance of these different formed elements before you look into some more histology slides. When studying the formed elements of blood, there's a few key features to focus in on to help you identify each one of these accurately. They can be quite difficult, and we talked about some of the identifying characteristics, but let's just focus on that a little bit more. First, you want to pay attention to the size and the shape of the cell, as well as whether they have a nucleus or not. So those are going to be the first big things that you can see. Is it small? Is it big? Somewhere in between? And can you find a nucleus? For example, red blood cells, they're going to be smaller. They don't have a nucleus. White blood cells are generally quite large, and they do have distinct nuclei. Then, to distinguish among the different types of white blood cells, observe the shape of the nucleus first, and then determine if it has or does not have granules in the cytoplasm. This is how you're going to tell the difference between neutrophils, eosinophils, basophils, lymphocytes, and monocytes. And then finally, when using the models, pay attention to the color and texture differences. Models often use color coding and surface texture to represent granules or show how something is smooth or rough. And this helps to identify especially these different types of leukocytes as well. Here are just a few more visual cues to help you quickly identify these different elements. First, the red colored structures are almost always representing red blood cells or erythrocytes, right? And they're red because these are the oxygen carrying cells, and they're typically the most numerous in any blood sample. Then if you see large cells that have lobed nuclei, you're most likely looking at white blood cells. The number and shape of the lobes is going to also help you to narrow down which type of white blood cells, like neutrophils with multi-lobed nuclei versus eosinophils with bilobed nuclei. And then the tiny little fragments that are scattered around among the cells, that's going to be the platelets. Another name for that, by the way, is thrombocytes. Thrombocytes are platelets. Leukocytes are white blood cells. Erythrocytes are red blood cells, right? The thrombocytes, the platelets, they're not full cells. So that's why they're the smallest fragments that you see in the cytoplasm. For lab exercise 2.2, you will be looking at histology images as per usual. You're going to need the lab exercise 2.2 table for this. This is again going to include helpful figures and descriptions to help you identify what you're looking at. You're going to want to identify all of the key formed elements based on their morphological features that we talked about and the reference images that are in the table. Specifically, you're looking at basophils, right, with the dark purple granules in the nucleus, the eosinophils with the large red-orange granules, bilobed nucleus, lymphocytes with large round nucleus and the thin rim of cytoplasm, monocytes, the kidney-shaped nucleus with the really big cytoplasm, neutrophils with multi-lobed nucleus and pale granules, and then platelets are, of course, the tiny little fragments scattered all around. Pay attention, again, to size, nucleus shape, whether or not it has granules, what color it stains. All of those things will help you to be able to distinguish these a lot easier. As you examine the stained blood smears, your main goal is to identify the formed elements with all of the visual features. This is a beautiful figure giving a good example of almost everything that you might see. See these erythrocytes with no nucleus and they're concave on both sides. There is the lymphocyte with a really, really big nucleus and very small amount of cytoplasm around it. You can see which of the leukocytes are agranular versus here's a basophil that has very, very dark purple granules. Here we have granules, but they're a lighter pink color. Okay. And then for the lab exercise 2.3, you're going to do an online simulation where you are blood typing, and it's going to walk you through, it's a click-through simulation where you will test to determine a person's ABO and RH blood type. So you'll go to the simulation link. The simulation will guide you step by step on how blood is tested using anti-A, anti-B, and anti-RH serums. You will observe the results, especially looking for agglutination, clumping, and then as you go through the simulation, take note of each step in the procedure and carefully observe your results for each step. Let's just quickly have a little preview of that simulation. So you start by clicking this little green button down here. It tells you a little bit of background information about the patient. You click next. Then you have your blood sample, anti-A serum, anti-B serum, anti-RH serum, and then you have the results here, and you will, answer the questions and use the results to determine which sample or which type you have in your sample. And that's going to be it for Lab 2. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics and leave your questions for me in the comments below.